Hello children. Today we will be looking at an experiment which is movement of water in animal cells. Okay. So in your previous classes you might have learnt what animal cell is. Okay. Now in this session we will be looking at movement of water in animal cells. Now in this experiment we need some materials which are 3 beakers, a petri dish, salt, dilute hydrochloric acid or toilet cleaning acid, 2 equal sized raw eggs, cloth to wipe, a strip of paper for measurement, okay, right, or here you can also use a measuring tape, okay, and 1 tablespoon, okay, 1 tablespoon, right. Now the first step in this experiment is we take dilute hydrochloric acid or floor or toilet cleaning liquid, and then into that we add our egg. Okay, we add our egg. Okay, now when we are adding egg into it, what will happen? The hydrochloric acid will dissolve the outer shell of the egg. You know that eggs are generally white in color, so that shell is getting dissolved because of the hydrochloric acid or the toilet cleaning acid. Okay children, then this eggshell dissolution takes 4 to 5 hours. Alright children, okay. Now after 4 to 5 hours, or when you observe that the shell has completely dissolved, okay, you will be left with a membrane and a fluid which is filled inside, which is nothing but your egg white and the yolk. Okay, right. So you take that membrane, okay, and wash the egg under tap water. Okay, you will wash the egg under tap water and then you will measure the circumference. Now when you measure the circumference, you can take a strip of paper or also at your convenience you can take a measuring tape as well. Okay, right, and of each egg with a measuring tape and mark it with a pen or a pencil. You just measure it and mark it with a pen or a pencil. Okay children, then in the next step what you do, prepare a concentrated salt solution in a beaker and place 1 egg in this salt solution and the other egg in a beaker containing tap water. So when we have these 2 eggs with a membrane, so we will place these 2 eggs in 1 highly concentrated salt solution and the other one in water. Okay, right. Now here one of the previous concepts will come into picture, which is the osmosis. Okay, right. Now we will look at the type of osmosis. Okay, let's go to the next slide. Now you can see that when we have put the eggs in beaker, now you can see that this egg has swollen in size. Okay, and this egg has shrunken in size. Okay, now this is the egg which is present in tap water. Okay, and this is the egg which is present in salt water, concentrated salt solution. Okay, now what has happened here? There has been osmosis which was occurring in these 2 eggs or egg membranes. Okay, now one type of osmosis is the endosmosis. Okay, and the other type of osmosis is exosmosis. So these are the 2 types of osmosis which are present. Okay, now let us see what is exosmosis. The process in which the water molecules leave the cell is called exosmosis. So why the water molecules will leave the cell? What is happening here? Because high water concentration is present, the water moves outwards. Okay, the water will move outwards. Okay, okay children, are you clear with this? Okay, and the next procedure is endosmosis. Now what is endosmosis? The process in which water molecules enter the cell is called endosmosis. Now when you see these 2 definitions, water molecules entering into the cell, that is high concentration of water, low concentration of water. So the water will move inside. So this is an example of endosmosis wherein the egg membrane has swollen. Okay, and the second one is X osmosis, where the cell has shrunk. And let me remind you one more thing, egg is a single cell, okay? Egg is a single cell, okay? So we can call egg as a cell. It is the biggest cell available, okay? Right? It is visible to our naked eye, okay? Right? So these are the types of osmosis available. All right, children. So I hope you have understood the class, okay? I will see you in the next class. Thank you.